The intricate dance between our gut and brain, once a whisper in the scientific corridors, now resonates as a symphony of profound biological significance. In the flourishing landscape of pediatric physical and mental health, a new frontier is emerging. Our earliest moments are surprisingly powerful, especially for the unseen world within us. Imagine a bustling inner world teeming with trillions of microscopic residents. It's not a science fiction, it's a reality. It's your gut microbiome and a lifelong health story. Hello everyone, I'm Hani Mkhater, Professor of Parasitology at Banha University, Egypt, and a committee and advisory board member of this conference. The third World Conference on Pediatrics, Neonatology and Infectious Diseases held in London, UK, mid-July 2025. Today, I'm going to talk about the unseen architects navigating the pediatric gut microbiome in health, disease, and resilience. With my colleague, Dr. Rida El-Badawi, she is a professor of hepatology, gastroenterology, and infectious diseases at Faculty of Medicine, and the executive director of the Center of Excellence for Studying Gut Microbiome at Banha University also. Below the surface of every child lies a bustling dynamic cosmos, the gut microbiome. Far from being a meager digestive aid, such ecosystem of trillions of microorganisms is a crucial performer of pediatric health, shaping everything from metabolic regulation and immune development, even to neurocognitive function. Recent discoveries revealed its boundless influence, renovating our understanding of childhood well-being and vulnerability. Unfortunately, disruption of this delicate balance during infancy can significantly alter this microbial blueprint. In order to provide a comprehensive overview of the topic, this talk delves into the fascinating, yet often overlooked, the gut's microbial inhabitants. Specifically, it addresses how dysbiosis of early life may predispose to allergic conditions, metabolic disorders, type 1 diabetes, inflammatory bowel disorders, atherosclerotic cardiovascular diseases, in addition to anxiety, depression, and a cascade of mental illnesses. Cheerfully, we will move beyond the bottle, exploring modern interventions and holistic approaches to nurture this vital garden. So, I invite you to join me as we discover how these first microbes build our lifelong health and well-being. Prepare to look at your inner world and perhaps your overall health in an entirely new light. Until the beginning of the 21st century, the neonatal gut was thought to be a sterile ecosystem, the sterile womb paradigm, with microbial colonization believed to commence at birth. However, recent findings have challenged this notion, revealing the presence of bacterial cells or DNA in the meconium, placenta, and umbilical cord blood from healthy newborns delivered via cesarean sections. During pregnancy, the mothers undergo several endocrine, immunological, and metabolic changes, creating an appropriate intrauterine environment, resulting in shifts in maternal, vaginal, intestinal, cutaneous, and even oral microbiomes. Over here, you could see the predominant bacterial composition of gut microbiome in infants, in vaginally delivered newborns versus cesarean-born infants. Could you see the difference? In the second table, you could see the composition of gut microbiome during the first 3 months of life of infant nutrition, leading to changes in the composition of gut microbiome in breastfeeding versus formula-fed infants. Following birth, the initial microorganisms that colonize the body of the infants are derived from maternal microbiota, including sources like vagina, skin, mouth, and stool, along with microorganisms from the immediate environment. In this chart, as you could see, factors affecting microbiome abundance and richness at the early stage of life, during pregnancy, parturition, and infancy periods. Imagine the earliest moments of your life as the groundbreaking for a magnificent structure, your body. Just as building strength depends on its hidden foundations, the microscopic communities that first settle within you, your microbiota, actively sculpt your immune system and fine-tune your metabolism from day one. But what happens if these crucial early constructions goes wrong? It can lead to imbalances, dysbiosis, that set the stage for future health challenges, like chronic inflammation, asthma, IBDs, and other immune disorders, which can be shown incredibly early, even before your birth. Okay, I could see that there is a gut secret language. But how early microbes whisper about allergies and asthma? It turns out, the way this internal universe develops can set the stage for common childhood battles, like allergic diseases, particularly atopic dermatitis, also known as eczema, and later asthma. These conditions are often silent whispers of an inadequate gut microbiome development and disrupted immune balance in that very first year of life. For infants who later develop eczema, an itchy skin condition, there is an increased abundance of certain bacteria like Clostridium and Escherichia coli, while beneficial players like Vividobacterium and Faecalibacterium were diminished. Studies revealed a less diverse microbial community in early life, plus depletion of bacteroides and specific Clostridium strains, directly correlated with their later development of eczema by their first birthday. On a hopeful note, a reduction in eczema severity during follow-up was directly linked to an increased beneficial butyrate-producing bacteria like Cobrococcus eutectus. The story continues with asthma, a condition influenced by genes, environment, and recently gut microbiomes. There is a growing credit for the gut's role in the perinatal programming of this respiratory challenge through the gut-lung axis, where the gut influences lung immune function both directly and indirectly. Dysbiosis occurring within the first 100 days of life could be impactful of this development of hypersensitivity disorders. It seems the unmicroscopic world within us, precisely balanced or disrupted in those earliest days, holds a powerful influence over our lifelong vulnerability to allergies and asthma. In this figure, you could see the bidirectional action of gut-lung axis. After exposure to antibiotics or drugs, the dysbiosis of gut microbiota lead to gut microecology imbalances. Bacterial structure components and metabolites of the gut microbiota, such as lipopolysaccharides and short-chain fatty acids, can regulate the development of asthma. Respiratory diseases in turn can cause intestinal damage, creating a vicious cycle through inflammation throughout the body. In this table, you could see the microbial imbalances associated with increased risk of asthma. Uh-oh, watch out, mothers! It turns out, the delicate balance and function of gut microbiome are profoundly linked to our metabolic health, specifically concerning obesity and its related challenges. In fact, our gut microbiome can even influence how our bodies use energy, potentially guiding us towards obesogenic microbiota that predispose us to weight gain. The whole story begins in infancy. What we are fed, a mother's own weight, the mode of delivery, the permeability of our intestines, early infections, and even exposure to antibiotics can all subtly direct the development of our gut microbiomes, setting the stage for obesity risk down the line. Human studies had started to reveal patterns, suggesting that our early microbial profiles might even predict overweight in children. Overweight 7 years old often have more bacteria from the Bacillodà phylum and fewer beneficial Bifidobacterium species. Amazingly, levels of specific bacterium, Bacteroides fragilis, at just 1 month of age has been strongly associated with the higher body mass index in children. Beyond the sheer numbers of microbes, there is another fascinating layer: epigenetic shifts. These sudden changes in how our genes are expressed, affected by our early environment and gut microbiome, play a great role in how obesity is programmed in our bodies. Okay, I could see, but how early microbes whisper about type 1 diabetes? Type 1 diabetes is a chronic and autoimmune condition where the body's own system mistakenly attacks and destroys the insulin-producing cells in the pancreas. Microbial investigation in children who develop type 1 diabetes reveal a distinct pattern. There is lower overall microbial diversity and noticeable shift in the balance between major bacterial groups, specifically Bacillodà and Bacteroidota phyla. Furthermore, levels of crucial beneficial bacteria, Faecalibacterium prausensii, a producer of protective butyrate, are often diminished in these children. In a diabimmune study, which examined children with a genetic predisposition to autoimmune diseases across Estonia, Finland, and Russia, the children who later developed type 1 diabetes showed a reduction in gut microbial diversity and elevated levels of specific bacteria, as you could see in the first row of the table. In a compelling study, researchers looked at 33 infants already at risk of type 1 diabetes and found a similar story, as you could see in the second row of the table. Even more telling, these children who went to develop type 1 diabetes also exhibited high level of human beta defensin 2, an antimicrobial molecule produced during inflammation, almost as if their bodies were already signaling an ongoing internal battle. Imagine a hidden battle raging within the gut, a hyperactive immune response mistakenly attacking its own host. This is the reality of the inflammatory bowel diseases, IBDS, a group of chronic conditions including Crohn's disease and ulcerative colitis. Both are characterized by persistent inflammation, notable decrease in the microbial diversity, and a lower abundance of beneficial bacteria like Rosebaria. Adding to their complexity, these diseases have strong genetic bases. What particularly challenging is that these debilitating diseases can first emerge in childhood and adolescence, setting individuals on a lifelong journey of chronic relapsing symptoms. Studies strive to understand the earliest whispers of the challenging IBDS and point to early exposures, such as antibiotics and even cigarette smoke during the fetal life as potential triggers. Interestingly, even breastfeeding has been explored for its potential role, though the full picture remains unclear. A fascinating observation has emerged. Infants born to mothers with IBDS often show an increased abundance of bacteria from the phylum Pseudomonadota and a decreased abundance of beneficial bifidobacterium during their first 3 months of life. The intricate dance between genes, environment, and our inner microbial world continues to reveal the secrets of IBDS. What? Atherosclerotic cardiovascular diseases? Yes, ACVD is a process of chronic inflammation where fats get entrapped in artery walls, leading to plaque formation. Conditions in infancy like premature birth, malnutrition, altered gut microbiome can quietly raise the risk of ACVDs decades later. What's more, certain bacteria like Pseudomonadota seen in preterm infants also found in adults with ACVDs. These microbes can damage the gut lining, fueling inflammation that spread throughout the body, which is a key factor of ACVD development. As you could see, the gut's hidden hand in heart health is a lifelong story. From the innocent world of childhood, a shadow stretches into adulthood. More than just hardship, early life stress encompasses abuse, neglect, and socioeconomic struggle, proving potent predictor for adult depression, implicating its severity and persistence. Early adversity can disrupt this delicate inner ecosystem, sparking a crucial conversation along the gut-brain axis. Studies reveal the distinctively altered microbiome in those battling depression with a specific bacterial group linked to depressive states, as you could see in this table. Amazingly, transplanting gut microbiome from depressed individuals into animals have even induced anxiety and depressive behaviors. Unfortunately, early adversity is consistently linked to cognitive decline in adulthood, impacting executive function, memory, and processing speed. These impairments stem from significant structural and functional changes in the vital brain region, profoundly affecting our stress regulatory machinery. The hypothalamic pituitary adrenal axis become dysregulated, leading to abnormal cortisol release. This in turn influences immunity, inflammation, and neuroplasticity. Children enduring trauma often face multiple psychiatric comorbidities, struggling with emotional regulation, social interactions, concentration, and persistent psychological distress. Now, here is a glimpse to the cutting-edge approaches offering hope. First, nurturing the gut garden. Modern interventions are focusing on targeted probiotic power. Imagine Bifidobacterium infantis and brevi like teeny delicate gardeners, precisely adapted to flourish the infant gut and process the beneficial sugar found in breast milk. Complementing this, prebiotic, specialized fiber acting as nutrient-rich soil with efforts mirroring human milk oligosaccharides to nurture these good bacteria. The ultimate strategy lies in symbiotic, a one-two punch combining these probiotic with their ideal prebiotic food, even exploring novel nanobiotics to ensure their successful establishment and enduring impact. Beyond the bottle, holistic approaches, vaginal seeding. Exposing the C-section baby to maternal vaginal fluid immediately after birth helps transfer beneficial bacteria. While promising, more research is needed to establish its safety and efficacy. Kangaroo care. Skin-to-skin contact between parent and infant has been shown to positively influence the infant microbiome by facilitating the transfer of diverse parenteral microbes. Mindful antibiotic use. Healthcare providers are increasingly considering the microbiome impact of different antibiotic choices, especially in vulnerable premature infants. The good news is breastfeeding can aid in the recovery of microbiome after antibiotic exposure. Dietary diversity and whole foods. As infants transition to solid foods, a diverse diet rich in plant-based fibers and fermented foods helps foster a varied and resilient gut microbiome. Finally, the uninvited guests who brought peace. Our modern ultra-clean lives might be making our immune system overreact, leading to chronic inflammatory conditions like Crohn's disease, ulcerative colitis, inflammatory bowel diseases, celiac diseases, multiple sclerosis, and asthma. Helminth therapy via introducing specific parasitic worms into the body, acting as trainers, gently re-educating an overzealous immune system, calm inflammation and restore balance, offering a counterintuitive yet promising new path of treatment. In conclusion, a child's gut microbiome is the unseen architect of their lifelong health. Modern factors often disrupt this delicate balance, leading to a lack of crucial beneficial bacteria, increasing the risk of physical and mental challenges. But this is not a story of despair, it's a call of action, brimming with hope. For within this teeny world lies immense plasticity and incredible capacity for resilience. Now we have powerful tools to intervene, targeted probiotic, prebiotic, and symbiotic, which could cultivate a robust gut. Additionally, practices like kangaroo care, mindful antibiotic use, and diverse diet further strengthen this vital ecosystem. Understanding and actively shaping the infant microbiome, especially during the first critical 1,000 days, allows them to build a fortress of health for our children. This proactive approach ensures they not only survive but thrive, setting them on a path to a healthier future. Rigorous future research, employing longitudinal studies and randomized controlled trials, precisely controlling for puzzling factors like diet and lifestyle, is essential to finally establish the causal relationship between our earliest microbiomal companions and our lifelong health and well-being. Thank you all for your attention. It's been a pleasure sharing these ideas with you today, and greetings from Egypt. We invite you to come and visit our beautiful country. This has been هانم خاطر.